Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Kevin Macaluso. He's the chair of Microbiology and Immunology at South Alabama University. We're going to talk about Ricketts and Rickettsiology. I didn't even know there was such a thing. So, Kevin, thanks for coming.
Thank you for having me.
Yeah, so what is Rickettsiology?
So Rickettsiology, it's the study of bacteria. These are obligate intracellular bacteria, and they were described over 100 years ago by a guy named Howard Taylor Ricketts, who was a Chicago physician who was sent out west to study this disease, and they didn't know the source of it. So he went from Chicago out west to Montana to the Bitterroot Valley and began to study this agent. And back at that time, they were calling it something like black measles or just spotted fever. And through a series of really nice studies with by himself and with colleagues, they were able to eventually identify that this was a tick-borne bacteria. And so people got it when they were exposed to ticks. And in the era of pre-antibiotics, it was actually quite lethal. So that was.
So it's a tick. So you get the condition's called Ricketts.
It's called the genus is Rickettsial, and the condition, the disease, has several different names depending on the agent. But it could be. But we basically call it spotted fever. Some variants are called typhus. Different types of typhus. It's a collective of disease causing agents, what we call Rickettsial diseases, and they can include like epidemic typhus, which is a louse-borne disease, murine typhus, which is a flea-borne infection, scrub typhus, which is a mite-borne infection, and we also have like anaplasma and Ehrlichial infections, which are all in the same larger group in the study of Rickettsiology.
Oh, okay. So these are tick-borne diseases. The tick will draw blood, and then these parasites are in the tick saliva, and they get into your blood. Is that the method that they get into you?
That's right. For the tick-borne ones, it's introduced into the vertebrate host or into the human host during the act of blood feeding. So yeah, as the tick is feeding, it will inject the Rickettsial into the feeding site. And that's basically the part of the puzzle that we study.
What happens once the Rickettsial is injected? What symptoms does it cause? What cells does it attack? What does it do?
Well, that's an active area of investigation. And so looking at it, if we just put the Rickettsial into a host, we know that macrophages are involved. So a lot of certain type of immune cells involved. It also infects the endothelium. When the Rickettsial get to a certain level of infection in the endothelium, which is the vessels lining the blood, it will cause them to essentially burst or leak. And so oftentimes with tick-borne spotted fever, the disease is actually associated with a rash, what we call a petechial rash. And that's just little breakage in your endothelium causing blood to leak out. So that's why I got the name spotted fever.
Okay, well, so it gives you this rash. That's, I guess, the visible sign. You get fever, it sounds like.
Right.
High fever. Do you get aches, chills, pains, any other, what's debilitating or bad about the condition?
So it's classic flu-like symptoms. So you get fever, chills, rashes associated with it often. One of the problems is with spotted fever, if it goes undetected as spotted fever, it quickly can spread through the body and you get organ damage. And, associated with blood flow, you also can get things like gangrene. So you can get something like loss of limbs. With certain species of Rickettsial, primarily the one that causes the Rocky Mountain spotted fever, if left untreated, death within 7 to 10 days will occur. So the mortality rate right now in the United States hovers right around 3 to 5%. This same agent causes diseases throughout the Americas, and in different parts of the world, such as Brazil, they have fewer cases, but the case fatality ratio can be up to 40 to 70% case fatality ratio.
Oh, wow. That's crazy. How is this diagnosed? When, how do people present when they go to a hospital or they go to their doctor? How do people find out that they have this problem?
Well, that's the problem. Is often is comes across as just a non-specific, flu-like viral infection. So it can be misdiagnosed. The rash is one way to look at it, and the real key for this is to inform a physician that whether or not you've had potential exposure to tick bites. That's going to be an important aspect of it. That can start leading them in the right direction. And then we have standard tests such as PCR, molecular tests, and as well as antibody tests. But for early on diagnosis, one of the key things is to also, include the fact that if you noticed a tick attached to you at any point in the recent history, or if you were outside hiking, you may not have noticed the tick, but it's other aspects like that to include in your visit if you go see a physician for this.
I gotcha. Okay. So what in particular are you studying about this? Are you looking to find better medicines for it, or are you looking to just understand the pathophysiology? Like, what are you studying?
So we study, our research in our labs mainly focused on the transmission kinetics or transmission biology, because you're dealing with bacteria that are transmitted by arthropods to vertebrate hosts. They form this kind of a triad of vector-borne diseases. And so there's a lot of variables associated with that. There's variables on the vector side. We know the vector responds to different rickettsial agents, and sometimes it can clear it, sometimes it becomes systemically infected and is able to transmit. We look at variables on the pathogen side, which include strain variation. So there's different levels of pathogenicity or virulence associated with different strains of rickettsia. And then on the host side, it's not as simple as the rickettsia get into a human host and make them sick, or a vertebrate host. It can be a little more complicated because they go through the tick, and we know that the tick has in its saliva, immunomodulatory or immune modulating factors that can essentially create a unique environment at that tick attachment site where the pathogen is deposited. So it's a complex interactions between these three species, or between these three organisms. And so we study really all aspects of it, specifically focusing on how the tick or any flea arthropod delivers it to the vertebrate host and what the pathogen looks like in that context.
So what the pathogen itself is what? It's a parasite, or is it what is it?
It's a obligate intracellular gram-negative bacteria. So these are all bacteria that we're dealing with here. And the obligate.
When you say is it inter or intra? Like it goes inside of our cells?
It's intra. It's an obligate intracellular bacteria. It literally lives inside of our cells, and it has to be in the cell to survive and cause disease.
Oh, okay. How does it gain entry then? Is it allowed in? Does it bind to a receptor like a virus and force its way in? Like, how does that happen?
It does. It does bind receptors on both the tick as well as the invertebrate in human hosts or vertebrate hosts. It will bind receptors using proteins that are expressed on its surface to bind a host receptor, and then it's endocytosed into the cell. It has a mechanism where it can escape a vacuole really quickly, and then it just lives in the cytosol, or the cell cytoplasm while it's in there. And it replicates in the cytosol, and that's where a lot of the pathogenesis comes from, is it gets to high numbers and then causes damage to the cells.
So it's very virus-like. It uses, may not use an actual spike protein, but it seems like it does.
But for a long time, it was considered, essentially, it was thought to be a virus because it gets down to whether or not it can be filtered out. So it is a lot like a virus, you're right. But
that's very interesting. What's the size of the bacteria, approximately?
About about 2 to 0.5 microns in length. It's relatively small for.
Well, it is, but could you use, I don't know the limit, the Abbey limit, but could you use light microscopy or at least fluorescently tag parts of the parasite and watch it enter? I mean, with viruses, they're in the nanometer scale, so you really can't if you use electron microscopy. But I wonder if you could use light microscopy and observe entry and observe, them escaping vacuoles and all kinds of stuff.
So the field has moved forward well with being able to generate mutants that express tags, so you can observe them in cells and you can observe their action. So we're about there on that aspect. Yes, we do that.
Well, that's really cool. So what have you seen it? Like, what have you observed, or what have you seen videos made by other scientists, and what does it tell you?
So the other videos are really interesting especially for spotted fever group Rickettsial, which is the tick-borne bacteria. They in vertebrate host cells, they utilize the host cell actin to move around in the cell. And there's several experts in the field that study just this mechanism, and it is impressive. There are videos of seeing these Rickettsial use the actual host cell molecules to move around, and then they also use this to penetrate into neighboring cells, and that's their mechanism of spread from cell to cell. So
why do you think a cell endocytoses this? It binds to a receptor. What is that receptor used for, and why would the cell endocytose it? Does it think it's food? What does it think it is that would cause it to do that?
It could. So two possibilities there. One is that it could go in there to endocyte. It will acknowledge it and endocytose it because it wants to destroy it. And so usually when something is endocytosed into a cell, it stays in a vacuole, and this vacuole is merged with the lysosome in the cell, and that forms a phagolysosome and usually destroys what's ever in the vacuole. So it's a way to get rid of microbes in a normal system. And for Rickettsial, they have this unique ability that as soon as they get in that first phagosome and are incorporated into the cell, they can escape that before the lysosome binds and before they get destroyed. So they've probably co-opted a natural defense mechanism of a cell to survive within the cell.
That's weird. I would think it would be very risky for a cell to endocytose something that it doesn't want in the first place, unless it's going to use it for resources. Like, I don't know if, have you compared it to other substances that get endocytosed and, a lysosome pulls up and binds, and what happens? It breaks the material down, but does it use it for, cellular constituents, or does it then just, have the vesicle go to the submembrane and be passed out as garbage, or what happens?
That's a good question. And so normally it's a process of just destroying foreign substances. Whether or not they're recycled, I guess, depends on the cell type. And
Has anyone tracked what happens once a lysosome empties its contents into the vacuole? And I guess it would be like, low pH type stuff that breaks down the bonds of whatever's in there. But then what happens to it? Has anyone followed the pathway and see where those things go? I forget the name you used. It's the combination of the lysosome and the vacuole. What are they called?
It's a phagolysosome.
Phagolysosome. Okay. Yeah. Has anyone traced the path of them once that's formed, where they go?
I'm sure they have. I guess I haven't really looked too much at that, so I'm going to step out of that area. There are bacterial pathogens that take advantage of the phagolysosome and take advantage of the low pH, as you mentioned, to become to activate them, and that's essentially how they live, and they can modulate the pH levels in the environment. There are other bacterial pathogens that can do that.
I was going to joke with you and say you don't know everything there is to know in biology. I'm ashamed, but you can't know. There's so much; it's impossible. That's okay.
It's that's why I stick with the arthropods primarily. Yeah.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So the bacteria is endocytosed. It's doing something to escape the vacuole, and then where does it tend to hang out? It just in the cytosol? Does it tend to go near any of the organelles? Does it seem to bind with any organelles or go to the nucleus, or
Well,
what does it do?
There are reports of it going into the nucleus as well, but it generally just stays in the cytosol. When you look at some of the other rickettsial agents, such as the anaplasma or the Ehrlichia, they literally live in vacuoles inside the host cell. So it's different biology depending on the organism, but for the spotted fever group and the typhus group, they'll live in the cytosol free and replicate. And that's why when you kind of keep going back to the word parasite, that's true. They essentially are parasitic on the host cell. They will use host cell functions and nutrients for replication.
So when they replicate, the particular ones you're studying, so they're not in a vacuole, they're hanging out in the cytosol. And then how are they recruiting? Are they stealing nutrients that are passing them by in the cell to the normal cell organelles? Are they going to be utilized? Are they, how are they co-opting the cellular machinery or nutrients themselves to grow and divide and replicate and all that? Any insights there?
There's been a lot of work on the biochemistry and physiology. I don't particularly do that, so I don't want to go too far into that area. We actually have other faculty in this department that do the nutrient scavenging aspects of rickettsia and how they use the host resources.
So what's your focus then of this whole pathway? What are you looking at? Again, the reason why I ask, I know I've asked you earlier, but I'm getting my head around it as I ask you. So that's why it may sound like a repeat. It's not like I wasn't listening. It's just contemplating the action of this. I have to ask you again, okay, now that I know this and listeners know this, now what are you studying?
So our focus is primarily looking at what the vector does, how the vector gets infected, how the bacteria survive in the vector, how they disseminate is the word we use. So once they get in, they'll come in with a blood meal. They would be in the gut of the vector. They got to get out of that and get in, infect the vector, get back up into vector salivary glands, and be ready to go into a new vertebrate host. I mean, that's the basis of a transmission cycle. So we study those kinetics primarily on the vector side, and specifically what the vector, how they get in, infect the vector salivary glands, and how they get down salivary glands, and how they get down or reintroduced into a new host.
So how does the tick feed? It bites, it penetrates the skin. Does it first inject saliva to prepare the wound site for the blood sucking? Does it have anti-clotting agents? What's there?
Tick saliva contains, we call them homeostasis modulators. So it has anti-clotting agents. It has vasodilators. It has to keep the blood flowing. It can suppress the immune response right at the site. It has molecules that suppress the itch response right at the site. So maybe that's why if you've ever been bitten by a tick, you would notice it. You really don't feel it or notice it all the time. You generally have to see them. And so it changes its profile because you likely know ticks stay attached for a really prolonged period of time, relatively for arthropods feeding. And so they secrete different types of proteins at different periods in the feeding period to throw off the host and to not cause a defense in the host that will block their feeding. And so a lot of times they create, depending on the species of tick, they can create a pool at the tick feeding site, and they can kind of modulate the host so that the blood keeps flowing to the tick during feeding.
So then how do the bacteria... So the bacteria are probably carried in that salivary matrix that includes all these factors, and that's how they get into the host? Or is there another mechanism?
That's for tick-borne rickettsia, that's the way they get in. So interestingly, for other rickettsia, for some of the insect-borne rickettsia, they actually get into a vector and then they pass out the gut through the vector feces, and it's those feces that are infectious. And those feces get rubbed into a wound, and that's the transmission cycle. So it's not necessarily through the arthropod feeding, but it's through the infectious feces.
Yeah, this one sounds more palatable. The other way sounds really gross, but it is what it is.
Yeah.
So
interesting biology for sure.
Yeah. Well, what happens once, like you said, people can die? Are there naive ticks that will then bite someone that has, this rickettsia in them and then get reinfected? Like, does the parasite make a circle or a cycle? Like, how does it return back to its, I guess, intermediate host?
So that's a really interesting part about our field is that, so for most of these bacterial pathogens of rickettsia, they literally live in the tick. The tick almost serves as a host. They are maintained what we call vertically, and so that's from mother to egg, and every subsequent stage after that. A tick has 4 life cycle stages: adult, egg, larvae, nymph, and then the nymph molts into adult. And the larvae and the nymph and the adult all blood feed, and if they acquired at any of those stages a pathogen, a rickettsial pathogen, they can transmit it to the subsequent stage. So we call that vertical transmission. So in sense, the tick serves as the reservoir and the vector for these pathogens, and that's fairly unique among the rickettsia.
Does it make the tick sick? And, can they live with them their whole life? Like, is anyone studying the ticks? Like, are they passed down when they breed, or like, what's their life cycle inside of a tick?
So yeah, that's a great question. And they do get maintained vertically, so they're passed down through multiple generations. And when you ask if the tick gets sick, that's a really great part of the ecology of this, because some of the rickettsial pathogens, such as Rickettsia rickettsii, is limited on how many times it can be passed through from mother to offspring to subsequent generations, because it does make the tick sick. Other rickettsial organisms that have been described in nature and identified, some of those are able to be passed 100% of the time, all the time, through every generation, and they're somewhat omnipresent. Certain ones, such as Rickettsial rickettsii, the one that causes Rocky Mountain spotted fever, it can only be passed in the ticks for a certain number of generations before it causes an overall fitness effect on the tick population, and then you don't see it anymore. So certain rickettsial pathogens require what we call an amplification host, which would be a vertebrate host, which a tick will feed on, become rickettemic, become bacteremic, and other ticks, naive ticks, can pick it up, and then that's how the organism is maintained in a tick population.
So what's, if your research is successful, I don't know what that means, we'll define it. What do you hope to figure out? What do you hope to accomplish in the near future, next couple of years?
I think if we can figure out what drives pathogenesis, what separates it, there's easily over a dozen rickettsial agents in ticks in the United States, for example, and worldwide there's many more. There's over 40 species of rickettsia that have already been identified. By studying these different levels of agents, these different virulence, these different rickettsia with differing virulence attributes or characteristics, and understanding how they move through the vector to the host, we can do a comparative analysis and try to identify a weak point, either A, just by determining what makes a pathogen, or B, identifying a weak point in this transmission cycle that could lead us to a point of intervention. If they need a certain molecule for infection or spread or dissemination in a tick, or they need a certain molecule to be transmitted, you can block that at either the pathogen level or at the arthropod level by targeting a tick molecule as well. So long-term is you identify a way to stop transmission.
Well, are there naive ticks that don't have these bacteria in them?
Yes, there are.
So how do you think the ticks get it in the first place?
Well, the mechanisms that we know for sure happen are, again, the vertical transmission. If they're naive ticks and they didn't receive it vertically, they can acquire it horizontally. And really interesting observation is that ticks can acquire rickettsia through co-feeding. So if you just have an infected tick feeding on maybe a rodent host, if a naive tick feeds in proximity to that, they can acquire the rickettsia during the feeding process. So they can get it through a vertebrate host.
Oh, wow.
Yeah.
Huh. Interesting. Oh, I didn't ask you, what cells in people does the rickettsia settle in? All cell types, blood? I mean, where does it go?
It's considered to be an endothelial cell infection, and so it targets endothelial cells in the humans. more recent literature has also described rickettsia infecting macrophages, possibly as a means to disseminate through the body.
But if it affects endothelial cells, then I would think that, it has access to most of the body then through the blood system. So I wonder if it sets up camp, proliferates, then, again, mixing language here, metastasizes and moves to another part of the endothelium, or if there's preferential areas in the body, it tends to settle and accumulate first.
It will, the current trend is going towards the idea that maybe it gets into macrophages first and uses that as a way to disseminate and then infects the endothelial cells. So it's using the macrophages as a way to move through the body. But it ends up being a systemic infection in humans for Rickettsiae.
Okay. I just didn't know how it's distributed in the endothelium, for instance, if it's uniform or if it's clumped or clustered in certain spots.
Oh, I get your question now. So yeah, it doesn't, it will start in one spot, but it will spread from there. And again, so these spotted fever have the ability to move from cell to cell. So it will spread out from certain foci of infection.
Okay, right. Makes sense. Is there anything that you could give to a person so that if a tick bites them, they essentially bite it back? They kill the bacteria inside the tick. I wonder if that would be a mechanism. If there's like a, sort of a vaccine that would also affect the ticks biting a person.
Not yet, but I think that would be one of the goals of the field, and is actively researched by a couple groups here in the United States and abroad. But right, the idea is if you could somehow, so I already told you that when ticks are feeding, they release these molecules that are essential to keeping blood flow constant and receiving the blood meal. So if you can block some of the tick molecules that are essential for that, you could prevent a tick from feeding. That's an active area of investigation for a lot of labs. And so, yeah, that's one approach, and that would be a really smart approach, smart way to go, because if you could block a tick from feeding, ticks are known to transmit several different pathogens, viruses, bacteria, true parasites. And so if you could block the tick from feeding, you would be able to block the occurrence of multiple tick-borne diseases.
Yeah, what would be cool is if you can make I know I'm going way off field, it just occurred to me, but if you can make a stent that had all the tick saliva embedded in it, minus the pathogens, so that you stent open someone's blood vessel, and then the tick compounds keep everything open and flowing, and prevent the stent from growing over. And I know there's no answer to it from you, especially, but because we're way outside the field. But the thought just occurred anyway. Harnessing tick saliva for good uses.
There's a pretty active area of research on that, and there's been some really nice work. It's the term is mining bugs for drugs, and yeah, you can identify vasodilators and other molecules in tick saliva that could be incorporated into human medicine. Yeah, it's not that far off the track.
Well, very cool. This is it's really interesting stuff. It's amazing, to study these creatures. So what's the best way for people to find out more about your work and get in touch?
We can look on the University of South Alabama website, and look through the publications. It's an active area of investigation, and it's an ever-changing field, and we're excited about the way it's going forward.
Okay, well, very good. Kevin, thanks for coming on the podcast. I appreciate it.
All right, thank you very much, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.